Hi everybody, welcome. It's good to be here with you today. We're going to be looking at a case study called The Mystery of the Seven Deaths. This is based on a true story, a really impactful one, in fact. And this is how we're going to proceed. You're going to take this case study one part at a time, and we're going to ask you to just look at part one first, read through it carefully, address the questions at the end to the best of your ability, write down or type up your answers to those questions. In other words, get as far as you can on your own, or perhaps with a partner or two. And only once you've done that, then you can resume this video. We'll talk about part one together and address the questions together. Then you'll pause the video again. You'll take a look at part two, complete the questions to the best of your ability, write down the answers, because you will be submitting those answers as part of this case study. Once you've done that, resume the video. We'll talk through part two and so on and so forth until we conclude the case. All right, so really do want you to pause the video, take your time. This whole thing said and done probably won't take more than an hour for you, and if you do really take your time, solve the problems to the best of your ability, look stuff up as needed, I think you're going to A, enjoy the case a lot more. It really is a really fascinating case, and B, you're going to learn a lot more and come away with this with a more developed understanding than you would if you proceeded in some other way. All right, so you're going to see a little screen in a second. Go ahead and pause the video, take a look at part one, answer the questions, and we'll see you in just a minute. All right, welcome back. So let's take a look at part one together. By now you know that there's been a spate of deaths in this major metropolitan city, and so let's just take the questions one at a time. Number one asks, are there any similarities or connections between these seven individuals? Well, there do seem to be some connections. For example, the symptoms are all very similar among these people who have died, and those are dizziness, confusion, headache, shortness of breath or rapid breathing, and vomiting. And the progression of the illness is also very similar. The deaths are rapid, occurring within a few hours of symptoms. Geographically, they are also linked in that all of these deaths were in the same neighborhood, happening around the same time. There is one further connection, though it's not universal as far as we can tell, and that's that the first victim took extra strength Tylenol, and these second and third victims, or actually third and fourth victims, had also taken Tylenol, perhaps to treat a headache. So we've got some connections here. The next question is, what questions would you want to ask the families to answer these questions? Well, we might like to know something about the medical histories of all of these people. Is it possible that they all have a pre-existing condition, maybe a cardiovascular issue or a vulnerability to stroke or something like this, which might explain what's going on here? We might also, because there's clearly a geographic link, all these people live within a close proximity, we might ask about their activities in the days leading up to their deaths. Could this have been a case of poisoning at a restaurant, for example? Did they eat at the same place or drink the same thing? Were they taking any prescription medications? Basically, we want to find out where they've been and what they've been doing, and also get a handle on whether they have any previous medical issues. The second question is, in your opinion, are these seven deaths connected? Why or why not? Well, you might just respond, well, we're doing a case study called The Mystery of the Seven Deaths, so yeah, probably they're connected. But silliness aside, we've got similar symptoms in the same place. We've got possibly this connection of Tylenol. They certainly do seem, at least on the surface, to be connected. But I guess we'll find out as we move on. So that was part one. Go ahead and pause the video again. Take a look at part two. Part two is a bit more involved, so please take your time with it. Feel free to refer back to notes or videos or your text in order to write down answers to the questions for part two. And once you've done that, you can unpause the video and we'll talk about it together. See you then. All right, folks, welcome back. We are in part 2 titled Autopsy Report, and here we've got a bit of new information coming from the results of the medical examiner's office as they conducted an autopsy and toxicology testing. So the first question says, recalling your knowledge of the function of organelles, what function of the cells was interrupted in these patients? Could this loss of function lead to the death of these individuals? Why or why not? So the organelle, which is mentioned explicitly in the autopsy report, is the mitochondrion, or the mitochondria. It says here, staining with specific dyes showed major mitochondrial damage within the affected tissues. So if the mitochondria were severely damaged, then we're looking at an issue with energy production, the synthesis of ATP to power all those endergonic cellular reactions upon which we rely for our moment-to-moment functioning and the sustenance of our life. If those most fundamental processes were interrupted, yeah, we wouldn't be surprised at all that that would result in serious illness or death. Second question says, given the data in the autopsy, were there any reports that seemed inconsistent with the immediate cause of death? Well, the immediate cause of death was determined to be hypoxia, which is suffocation or a lack of oxygen. Tissue sections from heart, lung, kidney, and liver all show massive cell death. Well, that's consistent with a lack of oxygen. Staining with specific dyes showed major mitochondrial damage. That could be consistent as well. We know the mitochondria use oxygen for the processes that go on within. This last one says oxygen levels in the patient's blood were approximately 110 millimeters of mercury, where the normal range is 75 to 100 millimeters of mercury. That means that the oxygen levels in the patient's blood were actually slightly elevated, which is awfully strange considering the immediate cause of death appears to be a lack of oxygen. So we've got a mystery here. How could it be that these people died of a lack of oxygen when they have plenty of oxygen in their blood? Well, maybe the next part will reveal some of this. So go ahead, pause the video, take a look at the next part, read it carefully, refer to your notes or whatever else you need to get a handle on it, write down your answers to the questions, and we'll see you in just a minute. Hi folks, welcome back. It's part 3. Things are getting serious. You know they're serious because there's a data table. All right, question 1 says, for each metabolite listed in the table, describe its role in cellular respiration. We're also going to answer the question of whether each metabolite is a substrate or a product, and we're going to address the main function of each of these. So the metabolites, which are all part of cellular respiration that we're looking at, are going to be glucose, pyruvate, NAD+, and NADH. Now there's other metabolites, but these are the ones we measured, and hopefully these will get us a little closer to an answer. For addressing these 4, glucose, pyruvate, NAD+, and NADH, we're going to go over to a diagram. And here we are. So before we start addressing the questions, let's just sort of walk our way through here briefly. So here we've got the net reaction for cellular respiration. A molecule of glucose reacting with 6 molecules of oxygen yields 6 molecules of water, 6 molecules of CO2, and of course a slew of ATP, somewhere between 30 and 40 molecules. Here we've got glycolysis on the left, the Krebs cycle or citric acid cycle or tricarboxylic acid cycle here in the middle, and then the electron transport chain and ATP synthesis occurring here on the far right. So the first question was, what are these molecules, what's their role in this process, and are they substrates or products? Well, the first one was glucose, and as we can see, glucose appears only once in this process at the very beginning. So it's a substrate, the key core substrate for glycolysis, and it's ultimately the source of chemical potential energy for this whole process. It's the energy, the chemical potential energy of glucose, which ends up recharging ADP and phosphate into ATP to power our various cellular activities. So glucose is serving as a substrate here. Pyruvate is a product of glycolysis and continues to carry a large portion of the potential chemical energy of glucose into the later stages of the process. So it's a product of glycolysis, but it's also a substrate for the citric acid cycle. The next piece that we're asked to look at is NAD+. And you can find NAD+ here as a substrate for glycolysis, here as a substrate for the citric acid cycle, and here as a product of the electron transport chain. So we see it in three places. We see it as a substrate in glycolysis and the citric acid cycle, and a product in the electron transport chain. And NAD plus is one of our energy carriers, one of our electron carriers. NAD plus is sort of the discharged version, so it is currently lacking the proton and the electron that it will pick up as it participates in these reactions. On the other hand, NADH, which is the final metabolite we're asked to take a look at, is the product of glycolysis. It's the product of the citric acid cycle, and it is a substrate for the electron transport chain. And NADH is the recharged version of NAD+. It's got an extra electron, it's got an extra proton, actually 2 extra electrons, and it's got an extra proton, and it's going to deliver those protons and electrons to the electron transport chain to power this pumping of protons across the membrane, which then can flow through ATP synthase to generate ATP from ADP and phosphate. So we've got a sense now, hopefully, of what the different pieces are and where they fit into this process. Let's take a look at the next question. So the next question says, are there any abnormalities in the levels of these metabolites in the victims? Develop a hypothesis about which pathway may be affected based on these abnormalities. So first, let's just see what's normal and what's abnormal. Glucose, normal. It's slightly different, but there's always going to be some variation. So we're going to call that a normal glucose level. This is also a normal pyruvate level. The NAD plus level, on the other hand, is quite a bit lower than the normal range. So we've got only 10 micromolar concentrations of NAD plus compared to 75 micromolar concentrations in the normal situation. So NAD plus, for some reason, is very low. NADH concentrations, on the other hand, are very high, some 8 times higher than their normal levels, 400 micromolar as opposed to 50 micromolar. So our glucose and pyruvate levels are normal, our NAD plus levels are very low, our NADH levels are very high. So what could be happening to decrease our NAD plus levels while increasing our NADH levels without affecting the amount of glucose and pyruvate? Well, let's take a look back at our graphic and see if we can figure out an answer there. So here we are back at our chart. We're going to try to figure out what's going on here and what pathway could be suffering that would explain the data that we have. The first thing that we might want to look at is glycolysis. Now, we know that the glucose concentrations are fine, so this isn't a matter of starvation or something like that. We also know that the levels of pyruvate are fine, which suggests that this process of converting glucose into pyruvate is going just fine. It's not increasing in its rate and it's not decreasing in its rate. We also find that the amounts of NAD plus are very low and the amounts of NADH are very high. So what could explain that? Well, could it be that the citric acid cycle is not functioning? Maybe. Well, it should be fine in terms of one of its key inputs, which is pyruvate, and it should be fine in terms of the ADP and phosphate, since the cell is constantly using up ATP and converting it back into ADP and phosphate. Now, we do know that the NAD plus levels are low, so it could be that there's a problem in the citric acid cycle that's leading it to consume more NAD plus, maybe. Let's take a look at the electron transport chain here. Now again, we've got an overabundance of NADH and an abnormally low amount of NAD+. Could something in here be causing that problem? Here I would say, yeah, that seems like a pretty good candidate. So if we know that the NAD+ is produced here, in fact, this is the only place in this whole process, really, other than fermentation, where NAD+ is produced. So if we have a very tiny amount of NAD+, and a very high amount of NADH, could it be that this step of the electron transport chain is somehow being interfered with, so that this conversion of NADH to NAD+ never happens? Well, that would be consistent with the data that we have that NADH is elevated and NAD+ is lowered. It would also be consistent with the fact that these people died. If your electron transport chains in your mitochondria are inhibited, you're not really going to be able to produce much ATP at all. You could use fermentation to make some small amount of ATP. It ends up being 2 ATP per glucose, something like that. But that's a very paltry amount compared to the 32, 36 ATP total when you can do this process. That won't keep you alive for very long. So right now it's looking like inhibition of the electron transport chain is the best explanation for what we're seeing so far. Let's take a look back at the questions. The last question is to explain your reasoning for your hypothesis, which I think we've done. So, final part coming up. Take a look, give it a read, and we'll be back in a moment. All right, folks, welcome back. Part 4, the role of cyanide. You're now convinced that you know the cause of death for these victims and quickly report it back to police, as this is a very dangerous situation. After realizing that the electron transport chain was no longer functioning, you started to suspect poisoning and ran a blood test for various poisons that you knew affected the electron transport chain. The test of all 7 patients came back positive for cyanide. Cyanide irreversibly binds to cytochrome C oxidase of the electron transport chain and prevents the transfer of electrons to oxygen, the final electron acceptor. Let's take a look at an animation that will make it especially clear how cyanide inhibits cellular respiration. All right, so by now you've seen this graphic quite a few times. Here's your electron transport chain, NADH, binding to one of the proteins here, giving up protons and electrons, protons being pumped across the membrane as the electrons are passed down the chain, eventually to their final electron acceptor, which is oxygen. Pumping of those protons creates a gradient so that they can flow back down through ATP synthase and be used to synthesize ATP. Here we're being told that cyanide irreversibly inhibits cytochrome C oxidase, which is this protein right here. So this diagram actually has a little thing where we can see directly the result of cyanide. So we're going to add cyanide to the system. So here comes cyanide. It's binding to the cytochrome C oxidase. Electrons cannot be transferred now from this protein to the oxygen. That oxygen is blocked. That also means that electrons cannot be transferred from this protein to the cytochrome C oxidase. They can't be transferred from this protein to this protein, from this protein to this protein, from this protein to this protein. That also means that this protein at the beginning of the chain can no longer remove electrons from NADH. And this is why the NADH amounts have risen dramatically, and also why the NAD plus amounts are reduced. Because that reaction by which NADH is oxidized to NAD plus can no longer occur. It's sort of like a traffic jam. You've blocked the off-ramp and the whole road has backed up. And if you think of this as sort of the on-ramp for electrons, no new electrons can make it into the system. No new NADH can be oxidized. Of course, the oxidation of NADH is just a means to an end, which is the generation of ATP. The real problem here is that by shutting down the electron transport chain, we are no longer pumping any protons across this inner mitochondrial membrane. That means we're not setting up a gradient across that membrane. That means there is no process that's going to drive protons down through the ATP synthase to generate ATP. This means a failure of ATP production and death. This is, at a molecular level, what is happening when a person suffers from cyanide poisoning. And depending on the dosage, this could be fatal within minutes, or it could be something that a person rushed to a hospital could recover from with some treatment. We'll talk about that treatment in a moment. Let's take a look at the next question. Number 2, given what you now know about the action of cyanide on cellular respiration, explain why the patients died of lack of oxygen while their blood oxygen levels were normal. Let's go back to the animation. So why did these people die, evidently, of a lack of oxygen when they actually had plenty of oxygen? Well, it's because this cyanide has bound to the cytochrome C oxidase protein, and it's preventing that protein from transferring electrons to oxygen. So there's actually plenty of oxygen, the final electron acceptor, around. However, that oxygen cannot be used. So it appears that the person has died of a lack of oxygen, but really what they've died of is a lack of access to their oxygen. And ultimately what they've died of is a lack of ATP. Question 3. Would artificial respiration or oxygenation have saved these people? Why or why not? So here, artificial respiration means putting someone on a machine that pumps air into and out of their lungs, and oxygenation means that you're adding additional oxygen to that flow of gases to try to increase the levels of oxygen in their body. Would that have helped? Why or why not? This is an interesting question because answering it depends on what kind of inhibitor cyanide really is. Now, you might remember from our study of enzymes that there are a couple different categories of inhibitors. And let's talk about competitive versus non-competitive inhibitors. So here's our model system. We'll say that blue here is representing the cytochrome C oxidase, purple or yellow, depending on which one it turns out to be, is representing the cyanide, and this little green triangle thing is representing the oxygen. Now, you may remember that in competitive inhibition, the inhibitor and the substrate compete for access to the protein. One can only bind when the other is not present. So if the oxygen's already bound, the inhibitor cannot bind, in this case the cyanide. What that would mean is if there were additional oxygen, we could occupy the cytochrome C oxidase with that oxygen and narrow the window of opportunity for the cyanide to bind to and inhibit the cytochrome C oxidase. In that case, additional oxygenation would be helpful to the patient if cyanide were a competitive inhibitor. If cyanide, on the other hand, were a non-competitive inhibitor, remember non-competitive inhibitors can bind to a protein even if the substrate is already bound and will still inhibit the reaction. If cyanide were a non-competitive inhibitor, then no matter how much oxygen there is, it's still going to be able to bind and inhibit that protein. And in fact, cyanide is a non-competitive inhibitor. So that means that if all of those cytochrome C oxidase proteins throughout all the cells in the body have already been inhibited by cyanide, increasing the concentration of oxygen will not help. However, if only some of them have been inhibited, increasing the concentration of oxygen may help. No, it won't save those proteins that have already been inhibited, but it may help the other ones to work more efficiently and rapidly and make up for the inhibition of the other ones. And in fact, in cases of cyanide poisoning, it's more common than you think. Cyanide poisoning is a common result of smoke inhalation. For example, in something like a house fire, they actually will treat someone with oxygen therapy, even hyperbaric chamber oxygen therapy, where they put you in, basically a steel tube and they provide very high pressure oxygen to get that oxygen into your blood at a very high concentration. And that does help and reduces the chance of death or serious consequences. So the answer to this question is sort of, it depends maybe. Cyanide does turn out to be a non-competitive inhibitor, so it's less effective than it would be if it were a competitive inhibitor. However, in a case of only partial cyanide poisoning, I think we'd be okay. That said, given the results that we have in this case study, that these people are dying with elevated oxygen levels, that suggests that they cannot consume any of their oxygen. They've been poisoned so severely that all of their electron transport chains are shut down. At that point, I don't think providing additional oxygen would be any help at all. Those people are doomed unless they receive an antidote that can react with the cyanide and remove it from the system. Next question. Final question is, looking back at the information you have about the people before they got sick, can you suggest a possible source of the cyanide poisoning? How should public health officials and police respond to this tragedy? So you may have deduced that it was all the Tylenol, and in fact, it was. So this was a real case. You can look it up if you'd like. It's sometimes referred to as the Tylenol murders or the Chicago Tylenol murders because it took place in the area, in Chicago, in the surroundings. What happened was somehow Tylenol capsules became contaminated with cyanide. They were put together, bottled, put on the shelves, and people bought them, took the Tylenol, and died of cyanide poisoning. We still don't know who did it or why. There was a man that came forward. He actually tried to extort the company that made the Tylenol and said, I'll stop doing this if you give me $1 million. The police caught up with him, but they weren't able to find any evidence that he had actually poisoned the Tylenol. So what they had thought was that he was going in, taking Tylenol off the shelves, putting cyanide in it, and then returning it to the shelves. So we're not sure whether he actually did that or whether he saw that there was an opportunity for him maybe to make some money and tried to take advantage. Now, he ended up going to prison for 10 or 15 years for extortion, but there was never any evidence to actually connect him to the deaths. So as of today, this remains a series of unsolved murders. It did lead to some changes. So product tampering became a federal crime with really steep penalties. It also, over the next 10 years or so, not immediately, started to lead to tamper-proof bottles, those ones we have to kind of like push down and turn, and also the little like foil caps and things like that. Prior to this point, those didn't exist. So it's partly because of these deaths that those kind of tamper-proof labels and bottles became a thing. After this, there were a number of sort of copycat crimes, something like half a dozen in the next couple of years. There was also a really high-profile series of murders carried out with the exact same method, poisoning Tylenol with cyanide, committed by a woman named Stella Nickel. She was convicted of the crime, Stella Nickel, and in the extensions for today, I'll put the story of Stella Nickel and why she committed those crimes. So this is the mystery of the 7 deaths. I hope it gives you a sense of how all these processes here are connected, how interfering just at one point, right here, can grind this whole thing to a halt and end up killing a person. This is a really fundamentally important idea when we're talking about metabolic pathways, this intimate interdependence, interreliance of all the parts. And so if you're starting to see that a little more clearly, then you're well on your way. Hope you have a wonderful day. We'll see you next time. Bye for now.